get my email regarding the final. Yes. Okay, good. So you know it's December 20th, and it's not at the same time as lecture, so it's actually an hour later. In this room at 3:30 p.m. From 3:30 to 5:30. So make sure that you have the correct time written down. Also, lab exams are this Thursday. So make sure that you're also ready.
We have a lab.
So any questions before we start? Alright, we're going to pick up with the digestive system. If there's time, we'll do the kidney. We'll see. Okay, so we left off talking about teeth. So we're discussing the various parts of the digestive system. And I want to talk a bit about how teeth develop. So basically, teeth are a combination of epidermis and dermis. They begin as epidermis and dermis, two outgrowths. The epidermal cells first produce what's known as an enamel organ. So it looks something like this. You can see the epidermis comes down and produces this enamel organ, which is this sort of C-shaped structure here. And at the same time, you have an outgrowth from the dermis called the dermal papilla, and that's going to grow upward and meet the enamel organ, and we'll see what happens with that in a minute. So the enamel organ grows downward at the same time as neural crest mesenchyme proliferates and gathers in the dermis to form this dermal papilla. The enamel organ then produces a specialized layer of cells, which we call ameloblasts. Ameloblasts produce enamel. At the same time, we have odontoblasts in the dermal papilla, which produce dentin. And it's actually the odontoblasts that induce enamel production in the overlying layer. So the dentin production by the odontoblasts also induces production of enamel in the overlying epidermis. Okay, so you should understand how this works. So basically, here's what this looks like visually. So we have the epidermis and the dermis here. The epidermis grows downward to produce this enamel organ, which is going to produce a layer of ameloblasts, which are these dark-colored cells in the middle here. And at the same time, the dermal papilla is produced by the dermis, and that produces odontoblasts. So you can see the odontoblasts here. And it's the dermal papilla that's going to induce production of enamel in the overlying layer. So we have simultaneous production of dentin and enamel. And it just continues to grow upward until the tooth erupts. So this is the process of tooth formation. And this is what an adult tooth looks like in mammals. So this is a human tooth, for example. So you can see what these structures look like. So enamel is this white porcelain-like layer that overlies the tooth, and dentine is inside, underlying that. And then there's what's known as the pulp cavity in here, which contains nerves and blood vessels. When your dentist talks about a root canal, they're talking about this canal that goes into the pulp cavity here. So this is called the root canal, and it's connected to the pulp cavity. So when we talk about the morphology of a tooth, there's the crown and there's the root. The crown is what's visible above the surface. The root is what's not visible. So the root is usually buried in the gum, below the gum line.
Is the tooth renewed? Like, is it always under construction like the bone is?
What do you mean?
You know how like osteoblasts and osteoclasts would break bone and then osteoblasts would fix it, remodel it? Does the tooth also get remodeled?
That's a very good question. I'm not sure the exact answer to that. I think the answer is no, actually. I think after the tooth is formed, it just remains. We are diffiodont dentition, so we have just the one replacement, and I don't think there's any change after that. So I think it's just, you get the new tooth and then it's fake. Okay, so teeth are important. Then the pharynx. So we talked about the buccal cavity and the parts of the buccal cavity. And now we're going to move on to the pharynx. What is the pharynx? So where's the pharynx? Yeah, the throat. So in terms of embryonic development, the pharynx is actually a very interesting area. And it develops into what's known as pharyngeal pouches. In fishes and some aquatic organisms, the pharyngeal pouches actually break through the surface to produce gill openings. But in mammals and most terrestrial organisms, they don't. So these pharyngeal pouches will form laterally in the developing embryo along the side of the pharynx. And they grow outward to meet external openings called the branchial grooves. So the branchial grooves, they're actually not openings until they meet, but they're in-pocketings from the outside. So the branchial grooves are basically moving in this way while the pharyngeal pouches are moving this way, and then eventually they're going to meet in the middle and there's going to be this open gill slit. There's a partition in the middle. So when we talk about the closing plate is what's separated them. So this here would be the closing plate. And when they do join, the closing plate will perforate, so it will form this opening. And you can see here is an embryonic shock. So if you take a look at this, you can see here are some pharyngeal pouches. This is basically I believe it's a frontal cut. So dorsal would be facing out toward you and ventral is facing into the cage. So it's sort of like a, frontal slice through the head. And you can see here are these pouches. So the pharyngeal pouches are outpocketings like this. And then you have these other in-pocketings here. And the branchial plates or the closing plates are in between. And there's an example of several of them which are already perforated. So you can see here in the spiracular one and also the second pharyngeal slit, the closing plates have already perforated. So it's an opening there, which will become the adult gill slit. If it's not an aquatic vertebrate, these just stay as pouches like this. And the fate of these various pouches differs in terms of what structures they become in the adult. So, okay, so in fishes and larval amphibians, the closing plates will perforate to form the functional gill slits. And in terrestrial tetrapods and vertebrates, they don't rupture. So if they do rupture, they're going to be sealed over after that. And there's actually a birth defect in humans where the pharyngeal slits will perforate and will not close. So occasionally a baby will be born with a small opening in the throat, and then that will be fixed surgically. But that's basically a remnant of our ancestry in terms of evolution. So it's like an ancestral gill. And occasionally people have a perforation in their ear, actually, also, that can be caused by that. So this would all be things that happen when the gill slits don't close. Okay, so this is a diagram showing you a lateral view of an embryo, typical vertebrate embryo, with these pharyngeal pouches. So you can see here, so there's 5 of them generally. So here's the first one, second, third, fourth, and fifth. And if this is a terrestrial vertebrate, they will just remain this way and then develop into adult structures rather than perforate. So this is what the pouches actually become. They actually become very many important structures related to the pharynx, the region of the pharynx. So the first pouch actually forms what's known as the tubotympanic recess. And that's going to surround the middle ear bones and become the eustachian tube. The eustachian tube basically connects your pharynx to your ear. And it's the reason why, when you're flying on a plane, you have to swallow on takeoff sometimes or chew gum to pop your ear so that it doesn't hurt from the pressure. So the eustachian tube basically connects your ear with your pharynx. The second pharyngeal pouch will become the palatine tonsil. There's a couple of tonsils in the pharynx. The palatine tonsil, the lingual tonsil, and where's the other one? Pharyngeal tonsil. So the second pouch becomes the palatine tonsil. Then the third and fourth pouch will contribute to the parathyroid gland and also the thymus gland. So 3 and 4, it's parathyroid and thymus. Pouch 5 becomes specifically calcium-regulating cells in the thyroid gland. It's not the only part of the pharynx that becomes the thyroid gland. So we'll see the floor of the pharynx also becomes part of the thyroid. So most of the thyroid comes from the floor of the pharynx, but the calcium-regulating cells come from pouch 5. Then the roof of the pharynx becomes the pharyngeal tonsil, and the floor of the pharynx, in addition to the thyroid, also becomes part of the tongue, the lingual tonsil, and the lung buds, basically. So lungs will bud off of the pharynx, ventrally. Okay, so we talked about the buccal cavity, we talked about the pharynx. We're going to move on to the alimentary canal now. So as you can see here, buccal cavity, pharynx. Alimentary canal is everything posterior to that, so the esophagus on down. There are 4 layers of the alimentary canal. So it's basically a hollow tube, and it forms from endoderm in the embryo, and basically forms as this internal tube that then differentiates. So the 4 layers of the alimentary canal are what we call the mucosa, the submucosa, the muscularis externa, and the adventitia. The innermost layer, which has the epithelium that lines the lumen of the intestine, is what we call the mucosa. So this innermost layer here. And it contains thin, smooth muscle fibers of what's known as the muscularis mucosa. So the muscularis mucosa is the muscle layer of the mucosa, separate from the muscularis externa. And then in addition to epithelium and the muscularis mucosa, there's also a thin layer of connective tissue between the lining of the mucosa and the muscularis mucosa. So in between the epithelium and the muscle is connective tissue. Okay, so that's the mucosa. Then the submucosa is inward from that, and that consists mostly of loose connective tissue and also autonomic nerve plexes. So the nerves and also some loose connective tissue. Then the third layer is what we call the muscularis externa. And when we talk about the smooth muscle of the intestine and the alimentary canal, this is what we're talking about. So it has an inner circular layer and an outer longitudinal layer. And basically, these are the layers that are responsible for peristalsis. So the waves of muscular contraction that move food through the alimentary canal. So that's the muscularis externa, the outer muscle layer, which controls peristalsis. Then finally we have the adventitia, that's the outer surface of the intestine. That's made up of fibrous connective tissue, and if there's a mesentery outside, then it would be called serosa. So an additional name if we have an additional layer. So if there's mesentery surrounding the intestine, then we call it serosa instead of just an adventitia for the outer layer. Okay, so let's take a look at what that looks like. So you can see here these four different layers. So we have the mucosa, which is the innermost lining here with the epithelium. And you can see here's the layers of that. So the innermost layer is this epithelial layer facing the lumen. And then we have this layer of connective tissue, and then we have the muscularis mucosae. Okay, and then we have the submucosa, which is immediately inward from the mucosa. So mucosa, submucosa here. And then external to that, we have the muscularis externa here. So inner circular, outer longitudinal. Maybe remembering back to intro bio where you probably got to look at slides of this. And then finally, the adventitia, which is called a serosa if we have a mesentery. So this is the serosa here. Okay, so you should know the four different layers of the intestine and the alimentary canal. Okay, so we have mucosa, submucosa, muscularis externa, and adventitia or serosa. Okay, so moving along to various parts of the alimentary canal. The esophagus, of course, is the tube that connects your pharynx with your stomach. It actually secretes a lot of mucus, which helps to coat the bolus of food and help it to slide down your throat. As food distends the esophagus, it actually initiates waves of peristalsis. So it's the stretching of the esophagus that causes peristalsis to begin. Okay, so you're not just normally undergoing peristalsis, but it's, when food goes in, it occurs. Okay, and then at the bottom of the esophagus, we have the stomach. The stomach is a primitively developed organ that serves to store food. So it begins digestion. It doesn't actually begin digestion, but it's the first part of the alimentary canal that does digestion. Digestion actually starts in your mouth with both mechanical and chemical digestion. I can't remember if I asked you this last week. Does anyone remember what gets digested in your mouth? Yeah.
Starches?
Very good, right, starches. So you have amylase, which is an enzyme in saliva, which breaks down starch. Okay, so carbs digestion begins in the mouth, but that's the only thing that begins in the mouth. So carbohydrates only. What begins digestion in the stomach? Anyone know that? Yeah.
Fats.
What?
No.
Fats?
Protein.
Proteins, right, good. So protein digestion actually begins in the stomach. The stomach also has some mechanical digestion as well. So it has, churns basically to mix up the food. It contains rugae, which are these folds on the inner surface that help to add surface area. So if you got to look at the interior of the stomach in your rabbit, hopefully you got to see the rugae. So it's these inner folds of the stomach that help to also provide a surface to aid with mechanical digestion. Okay, and the chemical digestion involves what we call gastric juice. So of course that's hydrochloric acid plus various digestive enzymes, primarily pepsin, which digest protein. There are three parts of the stomach. So we have what's known as the cardia, the fundus, and the pylorus. So the fundus is what we call the body of the stomach, if you're looking at that with your rabbit. The cardia, or the cardiac portion, is the upper part by the esophagus. And the pylorus is the narrow part at the bottom, which basically ends in the pyloric sphincter.